voice for a minute or two for you? Okay, that noise is pretty much the sounds of summer on the east coast of Australia, and the two lectures you've got today, one is on insect communication, so we'll be looking at a number of different means of communication, and the second is on Lepidoptera. So Lepidoptera is part of your lab this week, including holometabolous insect larvae, so from caterpillar to adult, and also you will be doing just Lepidoptera identification, holometabolous orders identification as well. So all those things. So this one's not so, but the next one does relate to the lab quite a bit. Okay, let's make a move. Whoops, I want to go on from there. What we're going to look at is a number of different means of communication. So we're going to start with sound communication, and then look at visual communication, and then the majority of insects are using quite dominantly chemical communication. So we'll look at that in just in the way they're communicating, but also look at it in a controlled context with both pheromones and allelic chemicals involved in chemical communication. So before I branch into sound, visual, or chemical, I just want to overview the whole context as I have there on communication. And we've got a number of different functions of communication that might be going on. One is disturbance, alarm, or distress. Second is, this is my space, keep out of it. Third one is agonistic, which is, I don't like you, or you're a rival. Agonistic calls. Then there will be ones that involve directly male, female sexual interactions. And the last one, which we won't deal with in this lecture because it is quite an extensive area on its own, is communication within social insects. So there's a lot of chemical and other communication, vibrational dances in bees, for instance, etc., for finding resources, but communication between castes and between queens, etc., in social insects. Now, given we've got these number of functions of communication, there is a number of ways that any one insect can actually change that to provide content that's meaningful. You can either change intensity or duration. You can combine signals, so you might not only just have a chemical signal, but you might have a sound signal involved, or two different types of sound signal at one point in time. The sequence that you are presented in is important to provide the information and the context. As you know, when someone says something in anger versus something jokingly, they have a very different context. So all of those things can combine to be able to give you a fairly good alphabet for insects to communicate. And the other thing is that it is quite a limited alphabet in some instances, like Lepidoptera, moths and butterflies, particularly moths, have a lot of chemical communication or pheromone communication, but it's dominated by 3 or 4 different particular chemicals. So they're mixing those up in all sorts of ways to identify different species. So it's not necessarily a vast number of resources that they're using, but all these variations can achieve it. Okay, so the first one I want to look at is sound communication. And insects do produce quite an amazing variety of sounds, but there are basically 2 approaches to sound production. One of those is that you hit some part of yourself against a substrate, which produces a sound, or the other is that you do it independent of substrate, so you're rubbing one or more parts of yourself together to produce sound. So you can do it either independent or dependent on the substrate. And the vibration frequencies are the things that you are rubbing produce the sound and the sound frequency. So acoustic signals, if you're producing sound, have quite a number of advantages over some of the visual and chemical. Firstly, acoustic signals travel well at work at night, whereas a visual signal wouldn't necessarily work so well at night. And it can travel quite great distances depending on the frequency. We'll talk about that as we progress through the lecture. Insects, though, tend to have frequencies that they call at more of the higher end of the spectrum because that's dictated by their smaller size. And we'll discuss how they get around that in the next few slides. Okay, so the first of the calling group that we want to look at are the mole crickets, or the gorilla talpots. Now, last week in the laboratory you saw a gorilla talpot burrow was there on the side bench. And basically it's an acoustic masterpiece produced by males to call for females. So they have they have two exit holes, so they're very hard to find. They escape pretty well, but they have what they call a tuning bulb here and flared horns here, much like a tuba is. And by the use of those flared horns, the width of the horn is about a third of the emitted wavelength to get the maximum amplification. So they call it around 2.5 kilohertz generally. What they position themselves in within this singing burrow is like this. So here's your acoustic bulb, and here are your wings which are producing the sound. And they act as a baffle so that the sound resonates in the acoustic bulb here and then eventually releases itself out through the horn through here, which is dug to a particular size and particular diameter. So it's a resonance bulb with a baffle, basically, and it increases the distance that the male's call will be heard over. So it actually gets to around 90 decibels, which is quite high, which is equivalent to standing about 15 metres away from the pneumatic drill. So it gets to quite high levels. And it also means that it can travel because of this increase in volume and acoustics of it. If it was on the surface, it would only be about 15 metres, but on this technique, it's about 600 metres that that call will travel. Although the female's ear apparently only hears about 30 metres, so they're sort of overdoing it. But the males do that to try and seem bigger, changing their frequency and their pitch to attract females who've got to fly there to mate with them. It's also quite noticeable if you've ever hung around lawns or pastures for a period of time after rain, or if you've just sprinkled towards the period of dusk, that they get pretty active as soon as they hear that. And if there is any dampness, then it helps with the acoustics of their burrow, and they can dampen it and make their acoustic bulb and everything a lot better. So they get pretty keen and excited when there's a little bit of dampness to build themselves an even better acoustic chamber for calling. So they're using the environment to improve their call and their burrows. Okay, we've got a few others that we also looked at in the lab. So one is the locusts and grasshoppers, the acridiids. Now these call by rubbing a part of their hind legs against the edge of the wing. And if we can persist with this for a minute, we'll get to there. It's a bit of an introduction of staying. And here you can see the male rubbing, it goes again, those legs over that wing edge, producing that sound. So it's not a particularly loud sound, and it's not as dominant as in some of the Orthoptera, but it does help communication, particularly in swarms. Bush crickets do something different, and you saw the bush cricket files and scrapers last week in the laboratory, and this is the bush cricket singing here. And you can see the wings involved, and I'll give you a slide for the next one. So this one, whoops, sorry. I'm going both at once now, get on to the next one. Okay, so this is field crickets, gorillas, which also do a similar thing, and this is the best illustration I could find to show this to you. So basically we have a scraper and a file. So this file has a number of teeth along it here, and that then moves across the scraper and produces the sound by the resonance frequency of the wing as it moves through that sound. So you can sort of get a rough idea of how high frequency their sound be by the number of teeth that they might have along there. Calling like that, then they'll wait. This is a female or male, but they have a burrow too, and females will be attracted to that burrow. In general, those sort of crickets are calling around 3.5 kilohertz. In terms of where we're at with calling, bush crickets can sing, or Orthoptera can sing anything below 10 kilohertz, around about to 6, up to about 100 kilohertz. Now our hearing range is only up to 20 kilohertz, so there's a huge amount, we think from 20 to 100 kilohertz, that they are calling that we don't even hear. Because they're quite small, they can produce much higher frequencies. Our voice is only 80 to about 800 hertz, so it's all below a kilohertz. So the advantage of being able to get a lower frequency, such as the mole cricket is achieving, or the lower frequencies like these field crickets, is that you get less attenuation or loss of sound as it moves through vegetation and other substrates. So there is an advantage to getting it lower, but they can reach very high frequencies that we can't hear. When you're out with students, you can pretty much quickly tell whether they've got hearing damage when something's around 20 kilohertz or more that they can't hear, and hearing damage is very common. Here are two bush crickets that I've taken photos of in southwest and western Australia. This one here is Tympanophora. This one actually is a predatory bush cricket, so it's unusual. Most, as I said, 91% or more of Orthoptera are herbivores. This one calls at a frequency of about 80 kilohertz, so you can only pick it up with a bat detector, but it calls very quickly, very fast, and then jumps and moves on to somewhere else. So it doesn't spend a long time in a period. And this one I put in here is to say that it's not always just the male taking the risk and calling. In some of the bush cricket species, such as this one here, Elephantodetta, the male calls and the female responds with a chirp. So there is a duet involved, and then he will move towards where he hears that chirp, call again, etc., and find each other via that methodology. A few little quirky things I wanted to mention in relation to it. One is snowy tree cricket, which is probably the most ubiquitous cricket in the United States. Sounds something like this. If you want to understand what the air temperature is, you count the number of chirps in 15 seconds and add 37 because they're, and they'll give you the degrees Fahrenheit temperature, because they're dictated by temperature as to how quickly they can move these wings and other things. So the other one, and I'm sure we've got students, some students certainly from China, is that there's an article in The Guardian a few years back now talking about a hobby that many people have in China, a dying hobby probably in terms of the older generations now, that they actually like to keep crickets beyond the autumn into the winter so that they can sing and remind them of the better times when it wasn't winter. So it's something that is done. They have their own little cages and feed them, and is quite revered for their singing. Another major acoustic communicator are cicadas, and some of those in the Hemiptera group in general. This is a photo of cicadas in Canberra. You can see enormous numbers of them on the one tree, literally standing under a rain of cicada pea underneath that tree. And what we showed in the previous lecture, just as a reminder here, that again it's independent of the substrate. They have a timbal, which is a membrane here, and a tympanum, which is the ear. So you can see the timbal here, and then a fairly hollow abdomen, which helps with resonance and increasing the sound and volume of the cicada. Some of them can reach intensities up to about 120 decibels. The greengrocer, it's around the Sydney area, and a jet engine is about 150 decibels, so they can get really, really loud with this. But there's an interesting group up in northern Queensland, the bladder cicadas, that have taken a slightly different approach to improving their sound, and that is they've actually made their abdomen an even bigger, larger sort of resonance chamber for improving the volume and sound of the call that they have. Another one in terms of communication is in some of the families of Lepidoptera, which we'll revisit again briefly in the second lecture. So in the Lepidoptera, we have a particular, well, noctuids, but particularly arctiids, the tiger moths, have also a tympanum, or a timbal organ, and a tympanic cavity. So they're quite capable of hearing and seemingly capable of producing a noise. For quite a number of years, the hearing one is definitely known to be able to pick up the sounds of bats using their echolocation, and if you put a certain frequency out, they'll drop straight to the ground to avoid being taken by a bat. But the production of sound was one that was curious, that was worked out in the last decade or so. So here is tiger moth, and this is the timbal organ here that produces the sound. And you can see that it's got quite an aposematic coloration, which is either oranges, reds, blacks, whites, which means don't eat me, I'm poisonous. And what they're actually doing is producing a call when they're flying, which the bats can identify. And they know that going for that is toxic, so they don't do it. So it's all like a little warning out there, a little radar warning, or a little warning all the time being broadcast that bats don't take me because I'm toxic. So that's what the communication both ways is going. So more or less interspecies communication between species, between bats and moths, although there is one moth group that does communicate using sound with its mating system as well. Another one here is in the Hemiptera as well, but with some of the Heteroptera. The green stink bug, for instance, uses a vibrational call, so it's moving its legs up and down on leaf surfaces. And it's an interesting study that was done a little while ago looking at the call of the males and females, where this stink bug exists on a large scale all around the world. And you can see here the male stink bug produces a very high frequency call in Australia, but if it's the one found in Slovenia, it's sort of not high frequency but pulsed, if you like, with much less frequency. And say in the female, Australian responds with a fairly long but amplifying call, whereas if you're in California it would just be a short staccato call. So they have actually evolved quite different signals as they have been geographically isolated, but they do use vibrational calling. The last one I want to illustrate with acoustics, before I just mention the applied end of the acoustics, is an insect known as the death watch beetle. Now it's called this, and I'll give you a sound here. I hope we're working away here well enough. So that's a sound that you wouldn't really want to hear. In the Middle Ages, certainly it became known as the death watch beetle. So when you're on your deathbed and everything's very quiet and your bed is riddled with death watch beetles, you can hear these little guys in the bed knocking their heads. They're basically headbangers banging their heads against the tunnels. That's the sound. So that's why they're known as death watch beetles. But certainly they're a wood borer, and they're putting something against the substrate to produce their sound. So it's a sound you wouldn't want to hear. Means something's not good coming along. Okay, in terms of pest management with acoustics, there's a couple of examples where pest management has been applied with acoustics. There is one up here which is, I think it's the green peach borer moth, with a colleague in Canada, Gerhard Gries, has been doing a lot of work in this space. And they've found that not only the chemical signal, but there is acoustic signals at close range when they're locating their mate. So if he provided just a pheromone, he would trap, X amount of moths, but if he had a pheromone plus an acoustic signal, he'd get far more of moths in that. So it'd be a combination of more than one signal that produces it. There is also means of trapping out things like mole crickets, which can become pests of pastures, but also of golfways, lynx and greens in particular. So you can trap them out with acoustic traps like this, or indeed use an acoustically orienting parasitoid to achieve something of that as well. But there was a paper only out a year or two back on insect vibrational signals as a new means of pest management, and they're looking at producing signals through vines to keep certain insects away with that vibrational signal. I think it's very experimental at this point, but it's interesting to see that sound is coming in a little bit more, but then there's a more expense to producing a sound than there is to producing a chemical. So it has a cost involved. Okay, let's have a look now at visual communication. And I want to just mention the organs that are picking up things in visual communication, and then talk a little bit about some of those things. So we have well, we've looked at insects. We've already looked at the head of the insects, and we've been looking at ocelli for a number of weeks now. And remember that you can see ocelli often arranged in threes like this, but sometimes there's two or one or no ocelli on the head. Now these ocelli are present, they're not in the larvae of holometabolous insects, but they may be reduced or absent on others. But they are on adults of a lot of adult species and hemimetabolous nymphs as well, if they do have ocelli. And the function of ocelli has been something of a debate for quite a period of time, and they've known not to have great visual acuity, so perceptions of light and dark are one of those things. There's a gentleman known, Eric Warrant, who did his original research back in Australia, but he now is based in Sweden, but comes back here every year and works on some of these things here as well, in Australian examples. And he's found that in some way it's to deal with flight motion processing and horizon detection, but has very limited resolution. But there was a paper only out about a month ago that also looked at the role of the ocelli in colour perception. So they're looking at honeybees, and they're saying, well, in certain environments a colour that's dark, a colour looks like this, in another environment a colour looks like this, the same colour. And they have managed to demonstrate that the ocelli actually can be a colour adjuster, so that they are actually perceiving the colour that they're always looking for by like a little corrector, if you like, to the colour that they're seeing through the light-dark sensitivity it has. So quite a few little roles, but we're only just starting to learn those. Now, if we're a holometabolous insect, we have stomata for eyes. These things don't look very impressive, and they're probably not. They don't have great visual acuity. They're probably very limited in their ability to see. If you think about holometabolous larvae, say caterpillars as an example, they're just sitting on a leaf chowing down. They don't have to do much looking for anything. They have made things eat. So here are your holometabolous larvae. Typically stomata are about 6, so we've got sort of 1, 2, 3, 4, 5, 6 here. Another one you can see here, and another one you can see here. So these are the visual organs of holometabolous larvae. Ones that we really want to think about, though, are the eyes of adult insects, and also their eyes are present in nymphal hemimetabolous insects as well. And they are made up of a whole lot of little hexagonal cells, or ommatidia. And in the break I'll show you a clip which is also on Milo on how that might work from what an insect may see. But essentially, again, visual acuity isn't the more important thing with ommatidia, it's motion perception. So as things move across each little ommatidial cell, then you get a change in the nervous firing to that, which then gives you a much better perception of movement than otherwise. So these are certainly different. So what I want you to think of when we're talking about visual, we're talking about sound, is that an insect isn't necessarily seeing what we're seeing, or hearing what we're seeing, or smelling what we're seeing. We've got to think of what their sense organs are doing and what they are seeing. So in terms of that, if we look at, well, one of the problems insects, some great cartoons around ommatidia. So, you know, if you go to the eye doctor, you spend a long time having to do the chart for each of your ommatidia, or putting your contact lenses in takes quite a few hours. However, what I am wanting to say is that if we look at the vision spectrum of insects versus us, they're already seeing something fairly different. So with the human vision, we're standing from 390 to 700 nanometers, which is the Roy GB, red, orange, yellow, green, blue, indigo, violet range. Okay, but with insects, they're shifted up a little. So they're not that interested in the red range as much, but they do get down to the ultraviolet range, both ultraviolet A and a little bit of ultraviolet B. So this means they're seeing something fairly different to what we're seeing. And they also have the ability to see polarised light. And the advantage of seeing polarised light is that in a clouded day you can actually see or detect the orientation of where the sun is, which then helps with navigation. A lot of insects use navigational cues for migratory flights or long distance flights. So the polarised light perception becomes important in there. Now we generally think of, when we computer screens and even our sight, we think of trichromatic vision, which is we have 3 different colour receptors, and the combinations of those colour receptors produce the colour that we're actually looking at. But there was a paper out 2 years ago now on Australian swallowtail butterfly, which found that they actually have 15 different classes of photoreceptors, which is the largest number of photoreceptors ever known. Previously it was 9. And the interesting thing about this is that they cannot see as clearly as human eyes, but are better at seeing a wide panorama, fast movement, polarisation, and grades of colour. So each of the cells may have a particular colour grade that it actually responds to, and if you map the compound eye of these butterflies, there is a concentration of some in certain areas and not in other areas, which relates to the functions that they're doing, whether it's something that's going to be underneath their feet or wider out. So they're not evenly distributed. So it's quite interesting that there is quite a variation within the compound eye in how they're positioned in terms of their sensitivity to different colours and so on. So if we're looking at insects with ultraviolet included in there, we can see some fairly interesting differences straight away. So here is a sulphur butterfly with ultraviolet. These would be brown, but with ultraviolet they look something like this. And here is the difference between this butterfly at normal and ultraviolet light. So we do have quite a lot of visual signalling in butterflies using ultraviolet colours. So that's one of the interesting things. But the other thing that comes to thinking about this then is that we the process of pollination and what flowers are doing to attract insects. And certainly there is chemical attractions, but there are also quite a large amount of visual attraction to reaching a flower and wanting to pollinate a flower. So if we look at flowers under what we see, this is what an insect would see. So under ultraviolet you can see there are a whole lot of guidelines here. So yep, you've got to come right down here to get the nectar and get the pollen forming. Again, dandelion just looks like that, but to an insect it would probably look something like this in terms of colour. And here is another flower that looks totally different between the two under ultraviolet and otherwise. So often we see a lot of guidelines that we wouldn't otherwise see, or contrasts in colours that we wouldn't otherwise see if we didn't have that ability to perceive ultraviolet. Concurrently with that, probably you could say that more of the red flowers are probably more pollinated by birds and mammals. Not so interesting to insects, but they do get up there, but the blues and the yellows are pretty attractive for insects. If we are trying to produce our own visual signals, these can be pretty good. You can use them at night, you can't see around corners, and distance is reasonable with visual communication, but there is a couple of groups of insects that I want to mention that do use visual, and this is one of them, which is a fly family known as the mycetophilids, and glowworms is the common name for them. So glowworms actually produce, well, I'll talk about it in relation to the next insect, which is what they produce, but as a larvae they sit in here, and you can see this is a mycetophilid larvae, and it produces this glow here, and it surrounds itself by sticky threads, and these sticky threads are what catches anything that is attracted to the light. So they're basically using it as an attractor to eat prey. So if you go to, say, Russell Falls at Mount Field National Park, underneath there are glowworms. There's plenty of glowworms in some of the caves, and New Zealand has a tourism industry largely around some areas where there is a lot of glowworms. So glowworms is one example, but the best example of something that is producing a colour or a flash at night. So these are operating at night in the case of fireflies, and they're not actually, trying to see if I've missed anything on notes, they're not actually flies, they're actually beetles. Okay, so we're looking at beetles here, and they are, in some parts of the states like Tennessee, North Carolina, spectacular displays, and certainly on my bucket list is something that I want to go see in some of the Great Smoky National Park, the Mountains National Park in Tennessee, etc. You get amazing displays. So basically the firefly in its abdomen has reflecting plates here, and it has compound known as luciferin, which with the enzyme luciferase, when it pumps oxygen into that with its trachea, glows with a beautiful colour there. And this has been used and exploited for identifying adenosine triphosphate ATP in molecular work as well. So here we have the lampyrosis there. So here we have a firefly, and the male's call. And again, by that whole thing about context and sequence, we can see here a number of different species. So this one has a flash, 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 and no flash for a period, and then a flash again. And this one's a constant duration between flashes. This one's a long flash on and then a long period off, etc. So we get the ability to have quite a number of species of firefly all calling into one area. Now what happens with them when they're calling, the female will produce a little flashback, and they'll recognise that flash and they'll move towards that female to try and mate with it, because basically they're busy doing that to try and mate. Now, as with all types of communication, with sound communication, producing a sound is dangerous, as we know. So a lot of those things that, like crickets and bush crickets, are calling at night when there are less visually hunting and less predators around. But in this case, with fireflies, there is one genus of firefly known as Phaturus, which is a predatory firefly species. And what it does is it mimics the female's call. It can mimic many species' call, depending on what's around in the environment. Pick that up, call as if it's a female, and the poor little male comes along thinking he's found a female and he gets eaten. Okay, and you can see here, this is an example of a really good display. But there is in different parts of the world, in Asia as well, but maybe from North America. So they're always eavesdroppers on signals. Okay, let's take a look now at chemical communication. I said chemical communication is probably the dominant form of communication in insects. And I want to just first again take a look at the receptors that pick up chemicals and discuss those in a more broader context. So we have two options for chemical communication. One is almost like a touch to the chemical, like a taste or gustatory receptor. So you would find these on maxillary pouch, on tarsi if they taste with their feet, like I said, flies do, and other areas like that. So they have a dendrite or hair which just has a single opening at the top, and then underneath that it has a number of nerve cells underneath there. And these nerve cells vary. There can be one that tastes, say, sweet, one that's sour, ones that's salt, so things. So it may vary depending on the insect and the particular diet, etc., that the insect might have. But they'll also use these to see if another insect has been on an area with marking trail and marking pheromones, which I'll talk about a little bit later. So it's not just food, but it is quite common in food. So this is labellum of a fly we've spoken about in week 1 when we looked at the external structure, how flies have modified their mouth parts to labellum, and you can see lots and lots of these taste receptors all over the labellum, which obviously is quite important for taste reception. The other sort of receptor that we can achieve to pick up chemicals is one that will be picking up chemicals in the air, volatile chemicals, chemicals that are floating around, okay? So these are smell or olfactory syncyllum, and here again we have the hair here, and we have the nerve cells going to dendrites up there, but we have, instead of just one opening, we have multiple openings along here. So you're increasing the potential for contact with volatiles going into the nervous sensor in there. So you've got a lot more, a big porous sort of site there, and again we have the same sort of thing down here with particular nerve cells being specialised in certain ways below there. I'm trying to find, do I have any more interesting notes to say that? I don't think there's any more. It's not always hair-like in how it appears. Sometimes it's plate-like. This is the antenna of a parasitic wasp, and you can see here this is again olfactory, but it's a plate-like syncylla there. as opposed to the more hair-like ones there. So it does vary in appearance. They're not all necessarily the same. Now if we wanted to try and get as much contact with anything that's floating in the air, then we do something like moths do, which have a very sexual dimorphism, but generally the male's trying to locate the female sex pheromone. So we've got some pretty impressive antennae here. And if you look at the sort of hairs on these are all designed for pickup of volatiles in the air. So these can pick up amazing, at an amazing level. I can't see where I've got this in my notes at the moment. I think it's about, it's in the billions that they can pick up in air. So it's very, very, very sensitive. I can't find the exact figure at the moment there. Where is it here? Maybe here. Okay. So females can release as little as 1 billionth of a gram per hour, and as little as 100 molecules of pheromone in a milliliter of air is enough to start a male in pursuit from quite a long way away. So the advantages of chemicals are quite good because you can use them in the day or the night. Is one obvious advantage, as I've said there. It works in the dark, yes, okay. It's effective around obstacles, and you can have flexibility in your transmission distances depending on the level of volatility of what you're producing. So it can be very short or very long in terms of distance, and it can persist for either a long or short period of time. So there's a lot more flexibility in chemical communication than there is in a flash of a colour or a quick noise that you might make. This is the original study back in 1959, which identified pheromones as important in insect communication. So we have the Japanese who ground up, and I think the number was something like 1 million virgin females to get 1.5 milligrams in those days of sex pheromone, which they labelled bombacol, of the silkworm. This is the silkworm. And their classic experiment was in a wind tunnel, and you have a little bit of filter paper with your silkworm pheromone here, and a female in a transparent box here. Fly them up there and they all go to the piece of filter paper and not to the female, showing that it's chemical and not visual that's important in their communication. We can do this a lot better now. We don't have to grind up 1 million females to produce anything like that, and we can synthesise obviously quite a lot of these pheromones. So we're a lot better off than 1959. This is the general pattern of a pheromone distribution in the air. So it's known as a pheromone plume. So here we have a female releasing the pheromone, and the further it gets away from the female, the more it distributes itself. Now you're going to have crosswinds and things interfering with it that break it up quite a bit. Now the way that a male moth orients in these is known as either a trawling or a tacking approach, or a zigzagging approach. So it moves upwards and downwards in and out of the plume to find the female. Now there is a couple of theories as to why that might be. Obviously if you have your antenna here and got the mouth not nice and wide, once you start to feel no chemical on one antenna and chemical on the other, then you would zigzag back in there, and that way you can actually find your orientation by moving like that. But the other possibility too is that they actually need to go out of the plume to almost empty their receptors of chemical overload and then come back into it. So the receptors might just be overloaded at that point as they're trawling through those pheromone plumes. But as you can see, the zigzag or tacking approach is the typical approach that they use to find the female in this case. Alright, I want to mention a few other uses of chemicals in a few other contexts. So we've briefly come across sex pheromones there, often produced by females to attract males. This one is another chemical produced by a female, but this time it's not to attract males, it's to tell males to piss off. Okay, so I haven't got a common name for this little ground beetle, but Pteristicus lupulenthes is it, and it produces an anti-aphrodisiac. So what this little ground beetle does is when a male comes along and wants to mate with her and she doesn't want to mate with him, she squirts him with an anti-aphrodisiac, which is essentially methacrylic acid, and it sends him into a coma-like stupor for about an hour to 3 hours, at which point the female has obviously disappeared and left him just lying on his back in his little stupor. So it's an anti-aphrodisiac that's trying to get rid of males from annoying females. But we also do have a possibility of an aphrodisiac as well. So here we have a male which has what we call hair pencils. In this case it's a butterfly. They have hair pencils extrude from the abdomen, you can see them here, and these have a number of chemicals associated with them, and they dust those on top of the female's antenna as they fly, and that forces the female to fly down, land, and open her wings up so that the male can mate with it. So it's his hair pencil aphrodisiac to encourage the female to mate with him. We have another couple of options that happen with pheromones in terms of what sort of things they're used for. And one is aggregation. So a great example of aggregation pheromones are in bark beetles. So here we have a number of different species of bark beetle, and this is their tunnelling underneath there. But if they're attacking things like pine trees, for instance, which use resin as a defence against being attacked, then there is an advantage to having a mass attack on a tree, so that you would then start on the tree, call out for others to come along, and collectively they attack that tree so that they can overcome the defences of the tree. And then you can use these pheromones, as you can see here, and collect bark beetles and control bark beetles with both aggregation and anti-aggregation pheromones. There are two more or three more that I want to mention in terms of pheromones. Now pheromones, when we're using the term pheromones, means intraspecific, so within species communication, okay? So alarm pheromones. In this case, we've got a couple of tortoises pursuing an ant, and I think we'll definitely get away. But the point from that reminds me is that a lot of alarm pheromones are used by subsocial and social insects. If you think about it, there's not much point sending out an alarm pheromone to help someone else who might get eaten when you don't want to get eaten. But if that happens to be related to you, then there's a much better option for you to tell all your relatives that there is something there. So it's social insects, ants, bees, wasps, etc. And quite a few aphid species as well, which are clonal and quite related to each other. You'll see the use of alarm pheromones to warn others that are closely related. You don't see it outside of those sort of subsocial and social groups. Another one that is used by insects, flies, and particularly parasitic wasps, is marking pheromones. So in this case, we've got a fruit fly who is coming along to a fruit, laying in that fruit, and she'll lay down a marking pheromone on that fruit to say that she's already laid in there. That'll tell other flies that there's already someone in there, go away somewhere else, or her, if she revisits that fruit, that she has already been there. In the case of parasitic wasps, used in a similar way, marking the host to know that it has already been parasitised. Of course, these things are then exploited by natural enemies of these that then find them through these marking pheromones. The last use of pheromones that I want to mention is trail pheromones. And again, trail pheromones is something that we see most commonly in social insects, because you're telling your near related kin that there is a food resource up there. So if you want a trail pheromone, you want it to be not lasting long. There's no point in it being there for 3 days. It's going to be very volatile and disappear very quickly. So here's an example of a fire ant worker laying a pheromone to the nest, and you can see within 100 seconds it's disappeared. But when you watch ants, they're always laying down that trail, and they're always following that same trail. So that trail pheromones lead them to the food resources. So certainly it is used quite extensively in social insects. Okay, I want to now just bring up some of the applications that are used for chemical communication. These particularly are around sex pheromones, and then I'll mention between species communication in the last slide. So for sex pheromones, you can use those in a number of ways, or any pheromone. It doesn't have to just be a sex pheromone. You can use it for monitoring. You can use it for mass trapping, for lure and kill, and for mating disruption. So for monitoring, there's a number of possibilities. You can use it to detect whether you get an outbreak, establishment of adult emergence. So when the adults first emerge, then you know that the eggs are going to be laid and the larvae are going to be there so many days later. You can map the distribution of a species, or indeed assess the changes in abundance from week to week. So important to have outbreaks. They have outbreaks if you've got a migratory species, you get the first front, you see that they're coming in, like some of the bollworm species, then you know that you've got something coming on pretty quickly. So all those are used for monitoring, and I'll just go to show you what sort of monitoring devices there are before I go back to this slide. Here's a number of different pheromones that we use for monitoring. This one is used for pine beetles, those ones that have aggregation and anti-aggregation pheromones. This one looks like the silhouette of a pine trunk. So they're attracted to that, and by the silhouette, and also by the pheromones put in there. This one's a very cheap form, a delta trap. I'm sure some of you have used these. It's just a wax board with a sticky surface in there. So it's good for some moths, small moths and things that will be easily trapped on it, but anything that's fairly large it's not so good for, because they're not going to be stuck on that sticky surface. So we go to something known as a bucket trap here. And this is the pheromone lure. It's generally a small piece of rubber scepter with the pheromone impregnated in it, and it's put into the lure. In this case here we're catching say large moth species or beetle borers and other things that are fairly large that wouldn't be trapped in this. And this is another version which is known as a Texas trap moth, which is a more open version of that trap. If we go back here, we've got some other options here for usage, and that includes mass trapping. So if we could, particularly if we've got a female attracting substance, that's really good because you can mass trap lots of them and kill them. It's not so useful to just kill males out of the population, it's more useful to kill females. Lure and kill is another approach, so you can get the females to come in, or the attracted individuals to come into a particular point, pick up some level of pesticide or biological pesticide that they take back to the area that they're associating with other insects, sort of like a Jonah insect if you like. And the other option is mating disruption, and I'll show you mating disruption here. This is an example of either a codling moth mating disruptants. You've probably seen these, you've been down the apples down in the Huon Valley. But these are twist tie lures, and you put them at certain spacing around the orchard, and then the hope is that that's flooded with sex pheromones so that the males can't mate with the females in those areas. They're completely confused by it. The problem is if you've got individuals coming from outside that are already mated, then you have problems. And so you need a fairly good understanding of the dispersal and the geography of the area to effectively use them, and the winds in the areas as to where they're going to be commonly pushing that pheromone. This is the use with trees and bark beetles of what's known as anti-aggregation pheromones. So you knock them on a tree and the beetles don't want to come to those, so there is a use in that. And the last couple of slides here is some work that I was involved with a while back now at Washington State University in Washington. These are growing of poplars. And there was a big industry for growing poplars there, but as soon as that industry started, then there was a lot of borers, and they had to develop a lot of pheromones for monitoring and managing it. This is a bucket trap for one of the beetle borers. This one here was successful. They plant their poplars as just cuttings, and then there's a mating, a beetle that attacks those cuttings, so they developed a mating disruptor, which is, you can see here, tied to these tubes, and they're all spaced along these areas here to try and stop that beetle from coming in and killing the newly planted poplars. And the last one, which we were looking at one point, was trying to see what the volatiles are coming out of the trees that the insects are attacking. So you'd find a tree where they're attacked and put a absorbent chemical absorber over that tree, and then try and dissolve that and look at the volatiles and try and develop something for controlling one of them, which was a large moth species that they hadn't yet correctly controlled. The last slide for the communication lecture is this one, saying that communication also occurs between different species, not just pheromones. And there's three names we have there: chiromones, alomones, and cinnamones. And a chiromone benefits the receiver, disadvantages the producer. So if you're a caterpillar feeding on a plant, you're producing, I think I've already mentioned this, or maybe I haven't, doing 4 lectures a week, so I can't remember which one's which, but the caterpillar feeds, it produces a volatile that attracts natural enemies. So it's a chiromone for that natural enemy. It's benefiting the parasitic wasp, but it's disadvantaged the caterpillar, okay? Because the caterpillar is producing something it doesn't want. An allomone, interestingly, confers an advantage on the producer by modifying behaviour of the receiver. That's a neutral effect of the receiver. And the best example of that comes in next lecture, actually. We're talking about a certain group, the coppers, butterflies, which live in ant nests, and they have a chemical mask, which means that they imitate other ants in the ant nest, and therefore they are neutral to the receiver, but so can hide in the ant's nests quite well. And the cinnamone benefits the producer and the receiver. So if you're coming from the plant's perspective, with that caterpillar example, from the plant's perspective, it's producing a volatile that it's benefiting it because it's getting natural enemies coming along and killing what was feeding on it. So a number of different combinations and possibilities in that. And that, I think, finishes that lecture. So we'll finish that. I've got a few clips and things for those of us